Last time the recording shut off randomly. I hope that does not happen again. Okay, I think it's lecture 8. So today we'll talk about insertions, mechanisms of inserting DNA. The other name for this is construct design. Okay, so the premise of this lecture is that is on transgenics. Like conceptually, how do you create transgenics? So what's a transgenic something? Really?
You've edited its DNA
By doing what?
by inserting genes that you want to.
Yeah, so transgenics is taking some gene X and then causing an insertion onto what? Insertion onto what?
Something else.
Like its body?
Well, like its genome.
Yeah, so it's chromosomes. Like, again, you have to start, I guess, visualizing what's happening at a molecular level. Because you could insert a gene onto different substrates. You could insert a gene into a plasmid or a virus. In general, when you create a transgenic organism, you're inserting some gene X onto that organism's chromosomes. Okay? And you can do this through various mechanisms. What's the simplest way to deliver a transgene? Simplest way to deliver a transgene that we've kind of already talked quite a bit about.
Plasmids.
Yeah, so, but there is a limitation to that. So the simplest way is you take gene X, you put it on a plasmid, and then there's a process where you get the plasmid into the cell. That process is called transformation. So you transform a cell with the plasmid, and now the plasmid has your gene of interest, and usually you can select for the plasmid then. So what's the caveat of this?
It's not actually in the genome.
Yeah, it's not integrated into the genome, that's for sure. That's one caveat. Another caveat is this only works for what type of organism?
Single cell prokaryote.
Yes, so exactly. So you can create GMOs with transformation of plasmids, but it only works for single-celled prokaryotes. Okay. This will not work for any animal, any multicellular organism. Why will this not work for a multicellular organism? There's probably a few reasons. Why would this not work for a multicellular organism? Take a guess. How many cells do you think are in a multicellular organism?
Hundreds of thousands.
Probably trillions. Trillions. How likely is it that you can take a plasmid and successfully transform a trillion cells with 100% efficiency?
Not very likely.
It's impossible. That never happens. That's why this is not really a thing that people do to create transgenic multicellular organisms. So in contrast, what do you actually have to do to create a multicellular organism? You first have to go to the stage at which it's not multicellular, which is what?
The zygote.
Yeah, the zygote. So you can either harvest eggs or harvest freshly laid eggs, which are just past the zygotic stage. And in that state, you can deliver DNA, and then usually you can successfully deliver that DNA to all the cells that derive in the body because they derive from that individual cell. So there is a way to create transgenic organisms that inherit some gene through the entirety of the organism, but you can only do that if you start at the single cell stage. Okay. So the simplest way to transform something is to construct a plasmid. Okay, let's talk about a second way. So a second way is so we talked about plasmids to transposons. So I'll briefly introduce transposons. There'll be a whole lecture on transposons, I think Friday or next week. Okay, so in a transposon system, does anybody know what a transposon is?
Jumping gene.
Yes, so in a transposon system, you create two plasmids. Plasmid 1, plasmid 2. Transposons are jumping genes. So it's a gene that codes for a transposase protein. So it's a gene with its own promoter. And it codes for a messenger RNA. It gets translated into a protein, which is called a transposase. And the transposase has an ability to bind DNA. And it binds to DNA at sequences that are flanking its own gene, okay? And so the transposase can grab its own gene and it cuts it out of the DNA and it carries it. So there's a complex of the transposon bound to its own gene and it carries it to a new site on the chromosome and it inserts it, okay? That's what we mean by jumping genes at a molecular level. It's a gene that codes for a protein that binds DNA sites that flank its own gene. It cuts it out, holds onto it, moves it, and inserts it in a new location. So transposons can be used as engineering tools to engineer genomes. So the most common way that you build this is with what's called a helper donor plasmid system. So on the helper, you build a plasmid that has the transposase gene. And what's been engineered is instead of the transposase having these sites that it recognizes, here I'm just denoting them with these circles, they've taken those out, okay? And they've taken those circles and they put them on a different plasmid, which is called the donor plasmid, okay? And in between the inverted repeats, the sequence that the transposon likes to bind to, you can put your gene X, okay? So you have two plasmids. You can also optimize this system where sometimes instead of having the helper donor system, sometimes this plasmid can also just carry the actual transposase itself. So you can make this one plasmid or you can do it with two, okay? Then what happens when you put both of these cells into a zygote? So let's say here's a zygote. How do you get plasmids like this into the zygote? Here's a zygote, a little cell, just a circle, but it's a cute little zygote. How do you get plasmids into the zygote? What would you do?
Would you use a vector?
What do you mean by a vector?
Like a virus?
A virus is not a way to deliver well, like there might be some virus that can deliver a plasmid. I'm thinking more physically, like just physically. How do you get these plasmids inside there? What would you do?
Can you give it to the parent that gives it to them?
You can do that, and we'll actually talk about some weird biotechnological engineering that can do that, but that's like way more complicated. Not right now. What's the simplest way to get these in here?
You injected it?
Yeah, you inject it. So you do what's called microinjection. So anytime you're gonna make a transgenic animal or multicellular organism, usually you're using microinjection, okay? So you have a little needle. It's usually a glass rod that's pulled in a machine that is microscopically sharp, okay? And you have this little microinjector and you load the plasmids in a buffer solution into the needle. You press a button and it spits out these two plasmids, plasmid 1, plasmid 2, into the zygote, and the zygote has many chromosomes on it. Probably should have drawn this bigger, but I didn't. Then what happens? Once you inject these, what happens? Once this is inside the cell, what's gonna happen?
Your transposase is going to bind to your other plasmid and then
Yes.
sit into the chromosome.
Very good. So the transposase gene, depending on what this promoter is, you probably have a promoter that's a constitutive active promoter, so it's always on. So as soon as it's in, it'll get transcribed. It'll get made into a protein, transposase protein. The transposase protein will not grab this because what it's looking for is its inverted repeats. So it'll find them over here and it'll grab this and it'll cut this thing out and it'll paste it. Here's zoom in. It'll then paste it somewhere randomly on the chromosome. Okay, so this is one simple way to deliver some gene X onto the chromosome. There's some downsides to this. It's random. It's a random insertion. So you're going to start learning that there are site-specific insertions and random insertions. What does that mean that it's random?
You don't know where it's going to end up.
You don't know where the landing site is. It's totally, well, it's not like there's probably some bias, some preference for some sequences, but essentially it's random. So you don't know what chromosome it's going to go on. You don't know where in the chromosome it's going to go on. So when this is randomly inserting, you can get what's called positional effects. So previously we discussed heterochromatin and euchromatin. What's heterochromatin? This is in the Euchs versus Probst lecture. Eukaryotes wrap their DNA, so what's heterochromatin?
It's when it's packed really tightly.
Tightly wrapped in the histones. So if your transposase inserts into heterochromatin, do you think you're going to get good expression or bad expression? Bad expression. You'll get low expression. In contrast, if your gene X inserts into euchromatin, which is loose, open, transcribed chromatin, you might get really good transcription, okay? So that's one example of a positional effect. Depending on where your gene inserted, you get better or worse expression, okay? Another type of a positional effect is you might have upstream some gene, let's say ribosomal protein, some subunit, I don't know, 25, just saying something random. And let's say this is really, really abundantly expressed. Sometimes this promoter can enhance the expression of this gene downstream. So just by the fact of inserting close to something that's really powerfully expressed, this could express at a higher level. That's another example of a positional effect, okay? So there are positives and minuses to random insertions, but just understand that one of the negatives is the positional effects, okay? Now, when you make a transgenic organism, you have to be able to recover this insertion. You have to be able to essentially like select for this insertion. So in this case, what we've inserted has no selectable markers. How could you modify the construct to put some kind of a selectable marker that you could select for? Which one would you modify, 1 or 2?
2.
2, you're gonna modify 2, that's right. What would you do to change it, to give selection?
Do you add a tag?
Where? And what tag?
A specific tag?
What tag and where?
Something on the beginning.
Right here?
Before gene X?
Before gene X, right here. What would you add? Give me something specific.
I don't know, like you said a histone tag a lot, but if you added that?
A histone tag. I've never heard of a histone tag. So let me say what's right about what you've said. What's right is that you have to add your selectable marker between these inverted repeat sequences that the transposon binds, right? That's correct, okay? You could add a marker. So instead of so there's 2 different terms here. There's a selectable marker or cassette, which implies like you're adding something that kills that you can select for, or you can, another term is just sort of like marker, is kind of colloquial. I know I put marker here and marker here. I'm sort of trying to teach you the colloquial language. A marker would be like if you had CFP. What's CFP?
Sign.
So it's gonna glow blue, okay? If you put that gene right there, and let's say you had a very strong promoter, and you successfully got an insertion, it's now gonna insert this. How would that change the phenotype of the organism you're trying to make transgenic?
It's gonna glow.
It's gonna be blue, yeah, okay? So sometimes what people will do is they will put something like an eye promoter. This is actually very common, especially for insects. They'll put an eye promoter of some fluorescent marker. And if that's the case, now how do you screen through your transgenic inserts? What are you looking for?
Different eye changes?
Yeah, you'll see blue eyes. You'll actually see blue eyes. And if it's not blue eyes, guess what? Your transposon didn't insert, okay? That's one example of a selectable marker that's not a cassette per se. Another common thing to have in Drosophila, so I'll show you how they do in Drosophila. So in Drosophila, there's a gene called white, okay? And the wild type white gene, the phenotype is red eyes, okay? And a deletion of white is, guess what color eyes? White eyes. So this is kind of confusing. In Drosophila, the genes are named based off their mutant phenotype. So when they discovered the white gene, they discovered it by a mutation that caused white eyes. So it got named white, okay? So a deletion of the white gives you red eyes. Did I spell white right? It's weird. Okay, anyway. If your zygote, if the genotype of your zygote is delta white, okay? And if you put the white gene between your transposon inverted repeats, what's the phenotype you're now going to select for after injection?
Red eyes?
Red eyes, you got it, yeah, okay. So it's kind of like annoying. I know it's like negative thinking. You have to kind of think it through. But when you make transgenic Drosophila, you're usually injecting a non-wild type strain, delta white, which has white eyes. And if you successfully get an insertion of the white gene with your gene X, the eyes turn red and then you immediately know. So you know in a population of flies which ones have your transgenic insert and which ones don't, okay? So this is an example of a transposon insertion and two different selectable markers that you could utilize to make a transgenic organism. Okay. Let's talk about another mechanism. So the reason that there are different mechanisms of insertions is because everything has pros and cons. And remember, the con of the transposon is that it's a random insertion. And you can get those positional effects which might cause problems for your goal or your experiment. So another way to do insertions is called homologous recombination. Okay, what's homologous? Similar what in this case? Similar DNA sequence. So homology is similar, but usually the biological example you hear is like, oh, the bat has the bones that the same bones that the whale has. That's homology. Yes, that's also homology. Usually when I say homology, I'm talking about the molecular sequence. So it means that the sequence of the DNA is the same. And recombination is just recombination. It's caused by or happens at sites of homology. Okay. So the way that you can do homologous recombination is if you have a plasmid, okay, and you put gene X, the gene you want to insert, okay, and let's say here's your chromosome. So we're inside the cell of the zygote, okay? This whole whiteboard is inside the cell of the zygote. Here's a plasmid. We want to get it to recombine with this chromosome, okay? A good thing about homologous recombination is that it's site specific, meaning you get to choose the site of the insertion, okay? So let's say this is gene A, gene B. Let's say we want to construct a insertion via homologous recombination between gene A and gene B. What should we do? how should we change this plasmid to get a homologous recombination between gene A and gene B? What could we do? How could we change this plasmid to cause that to happen? Remember, homologous recombination happens by getting those holiday junctions that I drew before. And those holiday junctions, the strand invasion happens at sites where the DNA sequence is the same. So what do we need in this to cause recombination here?
Do you need those?
What?
Gene A and gene B, flanking gene X.
That would be really cumbersome. You could do that. So like you could clone gene A here and you could clone gene B here, but that'd be really cumbersome to clone like the entire gene.
You just need like parts of each?
Yes, you just need what's called a homology region. So you're realizing that you just need a little sequence. Usually, like depending on the organism, usually it's like 25 to 50 base pairs, okay? So let's say we take this sequence here, let's call this homology region 1. Let's say the length is 25 base pairs. Let's say this sequence is homology region 2, 25 base pairs. And we can construct a plasmid where this is here and this is here, okay? And if we're working in an organism that has very efficient recombination like yeast, what will happen naturally is you'll get the strand invasion and this will automatically just recombine. So the symbol for recombination, the way that people draw it, let's say this plasmid was flipped. So I had 1, 2, gene X. The symbol for recombination is just an X. If you see this, that's implying that these two sequences are exactly the same sequence and they're creating a holiday junction, which then causes a recombination. And when a recombination happens, these DNA, it just swaps, okay? So the product would be gene X gets popped in here, and whatever was on the chromosome is now in the plasmid, okay? And so now you permanently inserted gene X into the chromosome. The reason I say permanently, in theory, this could happen again, but usually what happens is the plasmid doesn't replicate because origins of replication don't work in eukaryotic cells. Typically, like unless you're doing like a cell culture and you've got some funky stuff. But like in an organism, plasmids don't replicate. So the plasmid will disappear and this will remain, okay? Now in this case, we've got the same conundrum. We don't have any selectable marker. So how would you modify this to be able to select for this insert? Same as the last problem. Modify this to be able to select for it. Let's actually do a very specific example. Let's create a knockout in yeast of gene X. So we're going to create delta gene X. Here's the yeast chromosome. Here's gene X. And I want the knockout of gene X to be selectable by, this is in yeast. What selectable cassettes can we use in yeast? Give me one. Really? Selectable cassettes in yeast? There were like 5. Does anybody remember the process of selection in yeast? What's it called? Starts with an A. Auxotrophic selection. Okay, so in yeast you can select for things with auxotrophic selection. There's some options. Urethrin. Okay, good. Thank you. Very nice. Okay, so let's make it this insertion. Let's make this knockout selectable by Ura3 cassette. Okay. So we're going to make a knockout which means in this case, where do we want our homology regions to be placed from the chromosome? Where are we going to pull these from? These, where are these going to be to get a knockout of gene X?
On either side.
Yes, very good. Okay, so here's, okay, homology region 1. Homology region 2. Okay. We want the insertion selectable by Euro 3. So what should this gene be?
Euro 3.
Yeah, it should be Euro 3. Good, we're learning. Okay. If we construct this plasmid and we insert that into yeast with transformation, and then we make the yeast, we grow it in dropout media, so minus uracil minimal media, that will select for this insertion. This will recombine with this. This will recombine with this. Gene X will go into the plasmid, and depending on what the plasmid was, the plasmid will probably disappear, depending on what it was, and the insertion will be Euro 3. So now you've replaced that gene X with a Euro 3 selectable marker, and you can select for it by dropout media without uracil. So that's taught you literally how to construct a very simple knockout. Okay? So do you kind of understand now how inserting genes is entirely dependent on your ability to build and design plasmids? Does that make sense? Okay. Okay. That's good. Okay. Okay. How much time do you have left?
20 minutes.
Okay, good. We'll do another example. So, more ways to insert. So, transposons, random, homologous recombination, site-specific, but a con of homologous recombination is it's not efficient in some organisms. Okay, so homologous recombination works really well in yeast. So people have been doing transgenic knockouts in yeast for years, decades, because it's very efficient, okay? But it's not very efficient in mice. It can be done in mice, but it's not very efficient. It's not very efficient in more upper, like, multicellular organisms, okay? Here's a technique that can give you site-specific, CRISPR-Cas9 can give you site-specific insertions in multicellular organisms. We'll eventually have a whole lecture on this. For now, I'll just kind of like try to briefly throw it at you. Okay, so in this system, Cas9 is a nuclease. So a nuclease is essentially a scissors that cuts DNA. Okay. You can tell the Cas9 where to cut by what's called a guide RNA. So the scissors actually binds to a strand of RNA, which is called guide RNA. And the guide RNA is single-stranded, so it has exposed hydrogen bonds that are looking for other hydrogen bonds to bind to, okay? So just understand that Cas9 is like this little scissors that can cut DNA, that binds to a guide that's looking and scanning the DNA for that site, okay? That matches the guide. So it's site-specific because you can choose the guide. So let's say you want to make a insertion. You want to make an insertion into gene X. You want to insert into gene X, okay? So what you should do is you should make your guide match gene X, okay? So this is essentially a sequence that's going to match that, okay? So then where's it going to cut? Should cut right here. So you'll get a cut right here, okay? Once you have cut DNA, guess what the cell does to try to repair it? It's on the board. Yes. So what you should realize is that CRISPR-Cas9 essentially solves the problem of recombination not being very efficient because it can induce recombination by cutting the DNA at a specific site, okay? So if it cuts the DNA here, this DNA will kind of be like looking for ways to repair itself, looking for recombinants. And if you provide a template plasmid, so you still do the same thing, you've got homology region here, homology region here. Okay, and you probably have some, let's say the insertion you want to do is like GFP. You want to insert GFP. Now this plasmid can recombine efficiently specifically at that site because you've induced the breakage, okay? Any questions about that? I went over that really fast. I will talk about Cas9 and CRISPR much more later, but are there any questions about me just sort of like throwing all this at you? What you want to understand now is that there are different systems to insert genes onto chromosomes. They each have pros and cons. Okay.
So CRISPR's more efficient because there's already a cut.
You're causing the cut, yeah. You're causing the breakage, and because you cause the breakage, the cell realizes it needs to repair itself, and one of the endogenous repair pathways is recombination. So it causes recombination, and you can get an insertion then if you provide a plasmid. Yeah.
How much more expensive is it?
More expensive? This is not very expensive nowadays. What's expensive is the labor, like to do this. To pay a graduate student to do this stuff is very expensive. But the supplies, that's not very expensive. And nowadays there's many companies that will just do this for you. Like you'll design kind of like the construct and they'll just do it for you. Unless you're working on something real weird. Okay. So understand there's different ways to insert stuff. Let's think about, your first design considerations. So you should start to realize now that in order to get some insertion, you have to design and build a plasmid that usually will cause that. And the first thing you need to decide is the mechanism of delivery. So how are you going to do that? Are you going to use a transposon system? Are you going to use CRISPR? Are you going to use homologous recombination? Or are you going to give a plasmid, etc.? That's what I would call the mechanism of delivery. Okay. Let's also now talk about promoters. Okay. So when you insert something, if you are designing something, the choice of the promoter is a very important choice. Okay. So there's different types of promoters to consider. There's constitutive. What does constitutive mean?
It's always on.
Yep, always on. So constitutive promoters are always on. Things like actin, tubulin, myosin, ubiquitin, those promoters, always on, okay? There's inducible. Inducible is kind of like the arabinose system or the Gal1 system or the LAC system where you're adding some ligand or reagent and it's turning it on or off, okay? There's weak promoters. There's strong promoters. What other promoters? That's pretty much it. So just understand that as we move forward, we'll eventually be talking about very specific promoters, and we have already. Understand that there's these divergences. How much time do I have now? Okay. Let's talk more about design considerations. So let's talk about tags. Tags is kind of like a catch-all term that implies a lot of different things, okay? There's what are called affinity tags. Affinity tags, affinity in chemistry is it likes to bind stuff, okay? So an affinity tag would be something like a His6 sequence right in front of your gene. Here's a start codon, here's a promoter. A His6 sequence in frame, in fusion with your gene X. That's gonna create a protein X that has this little 6-histidine tag, and that histidine tag, that likes to bind metal like nickel or cobalt, okay? That can be useful if you wanna make a bunch of this protein and then you want to purify it, you've now constructed a specialized protein that you're able to purify by getting it to bind to beads. Okay, that's an affinity tag. Another affinity tag is GST. So instead of His6, there's a protein called glutathione S transferase. So that's where you actually have X, and you've actually put a whole nother tag, a whole nother protein, GST, attached to that, and GST binds glutathione. So you can buy beads that are coated with glutathione, and now your X will bind to those beads. So it's a way to purify. So remember that affinity tags are used to purify protein. You can have secretion tags. Okay, so that would be like a secretion signal, that if you have a secretion signal on X, whatever has this gene is now gonna make X and pump it out of the cell. You can have nuclear localization signals. We already talked about that. So if you have an NLS, in-frame infusion with gene X in your plasmid, it's gonna cause gene X or protein X to go to your nucleus. Fluorescent tags, we've already gone over those in the context of localization experiments. So maybe you put GFP, etc. Now it glows. Okay, so what I'm pointing out is that these are all things you need to think about when you're designing your plasmid. And every single one of these decisions, should I have a tag? Should I not have a tag? What's my promoter? What's the gene? What plasmid am I using? What's the backbone? All those are key, significant decisions that will make or break whether or not this will work. So you have to think through all those decisions, okay? Another thing you might think through is codon optimization. Okay, so remember back to the central dogma. You have a gene, and the gene makes messenger RNA, and there's codons, okay? And each of these codons represents an amino acid. How many amino acids are there? 20 amino acids. How many codons are there? How many codons are there?
6.
How would you actually mathematically, this is actually an interesting probability question. Does anybody know how to actually do the mathematics of it? How many, so a codon is 3 bases, right? How many bases could, in theory, be at position 1? 4. How many bases could be at position 2? What? And then in the third position, you could have 4. So this is how many permutations of codons you can create. So it's 64. So there's actually 64 codons, okay? Not all those code for amino acids, because some are stop codons, okay? But there's way more codons than there are amino acids, okay? And for each codon, there's also a tRNA. So there's about maybe 61 tRNAs, okay? All organisms have different preferences, what are called codon preferences, okay? So in E. coli, let's say, I haven't memorized the codons off the top of my head, but imagine there's 4 codons for glycine. So glycine, 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 glycine. If somebody wants to look them up, that'd be helpful, but I'm just making them up. These are probably not right. I'm just making this up. Let's say these were the codons for glycine, okay? And let's say E. coli has a preference where E. coli uses 75% this one, 10% this one, 5% this one, and then what would I need? 10%. 10% this one. So what you're realizing, again, this is not exact. These are not even the right codons and these are not even the right percentages. But what you're understanding, or you should understand, is that different organisms have different preferences for the codons that they like, okay? So if you were gonna construct a transgene that you were gonna give to E. coli, that you wanted it to express really well, you wanted it to make a ton of protein, which codon would you choose for most of your glycines?
The GCG.
Yeah, you'd want this one. You'd want to choose this one, okay? Because if all your glycines were coded with this, there's going to be the most tRNA for this, and so you're going to get high production of that protein. Whereas if you choose this one is called what's called a rare codon. If you have a ton of rare codons, your protein expression will not be very efficient, okay? So then this brings up what's called codon optimization, where you have a sequence. Here's your full sequence for gene X, okay, of your codons. You can change every single one of these codons and it can still code for the same protein. And so you can pick at each of these codon positions the most abundant codon for that particular organism. And so you can change the DNA sequence without changing the protein sequence. Does that make sense? Make sense? Okay, that's codon optimization. Are there any questions about that?
How would
So,
you do that?
what?
How would you do that?
How would you do that? So there's a computer algorithm. So what? Okay, so let's actually go through the long process of how you would do that. One, first you would need to know the preferences. How would you find out the preferences of the organism? What would you need? It's a really good question, by the way. How would you find the preferences of an organism's codons?
Maybe isolating a lot of yeast RNA
Would you make a
and
list?
seeing the most common.
No, first you just sequence the genome. So you would sequence the genome and you would look at all the genes in the set of that organism. So you'd sequence the genome, you'd identify all the genes. In those genes, you would identify all the codons, and then you'd make a tally of, you'd sum up how many codons there are of this one, how many codons there are of this one, and then you'd calculate percentages for all the codons. And then you'd be able to know that, okay, whatever organism it was, let's say it was Drosophila. Drosophila has these preferences based on its sequenced genome. Does that make sense? And then you would take your, how to actually implement that. So there's computer algorithms that know these preferences because the genomes have been sequenced. And so you tell the computer algorithm, take this DNA sequence and just change all the codons to the most abundant codon. That's codon optimization. And then you just get a new sequence. And that sequence can then be synthesized and put into your plasmid. So the whole point of this is that you have to think about what organism are you giving it to, whatever your gene is. And you have to make sure that your construct has been optimized to be given to that organism in the first place. Otherwise, it won't work. It won't work very well. Okay. We'll talk more about this stuff later. How much time do I have?
5 minutes.
5 minutes. Let's do, okay, so here's an example. So here's an example question. Is, let's say, this thing's already not working. So here's an example, so bad at that. Here's an example test question. Okay. Let's say this is an agarose gel. It's an example. Let's say you run an agarose gel and you're trying to do a restriction enzyme digest, okay? And you're trying to check to see if a plasmid has your insert X. Okay, and let's say, I should draw this a little bit different. Let's say the insert size is 1.5 kb, and the backbone size is 2 kb, okay? 1.5 insert, 2 kb backbone. Here's your ladder, here's let's say your ladder looks like this. And you do your restriction ends on digest, you cut your insert out, you want to check to see if it's there. So your backbone, your negative control is run in this lane, and you see this. Here's your backbone. And then you run your test experiment, say sample 1 here, and you see this. In theory, what should you see? You should see what? You should see 2 bands in theory, because 1 is 1.5, 1 is 2. So in theory, in a perfect experiment, you would see something that looked like this. That'd be a perfect experiment. But in reality, sometimes things are not perfect, and you see this, you see a smudge. What should you do in this case? What's the problem? This is a very common problem in agarose gels. First, identify what is the problem.
The ladder
What about the ladder is the problem?
is too condensed.
Yes, the gel is too squished. There's not enough separation. That's the problem. So yes, exactly right. Like the ladder, this pattern is telling you that it's too squished, not enough separation. So how do you solve that problem? What are things that you could do to solve that problem?
Longer gel.
Like make it longer?
Yes.
Although usually that could be work, but that's usually problematic because usually you run these in like cast setups where you can't change the size. So how else could you fix that? But that's a good answer.
Could you change the composition of the gel?
Yes. What would you do to it? That's exactly right. What would you do to it? So you change composition. It's made of agarose. So essentially you can either add more or less agarose. Should you add more or less? I'm trying to think. If let's say
If you make the holes in the matrix smaller.
it should be, yeah, so you should bump up the concentration of agarose. Okay, that's gonna make it harder for the molecules to run through. And then in that new gel, you should get a better separation of the ladder, and then you should be able to see, I messed this up, did I? No, I didn't. Here's 1.5, you'd see the backbone, and then you should see this. So that's a good answer. So you can change the concentration of your agarose gels so that they look better or worse.
Is 1.5 the backbone in that?
Oh, I'm sorry. Yeah, 2.
Okay.
So here's backbone. Here is your successful insertion.
Okay.
Yes. Good answer. Okay. That's it. Just I try to throw in like examples so that where I put something on this in the test, you have seen it. Okay. Good lecture.